Welcome to the PubMed Daily Review for April 24th, 2026. Today we scanned 5,771 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, a trial of hypoimmune allogeneic CD19-targeting T cells in refractory systemic lupus erythematosus by Vanya Ivkovic and Augusto Vallo. Let's break down its key contributions. Chimeric antigen receptor, CAR, T-cell therapy is a promising avenue for treating severe autoimmune diseases like lupus. However, current approaches have significant hurdles, including high costs and the risky need to temporarily halt immunosuppressive drugs. This study explores a novel type of CAR T-cell called STAR-T, designed to be more sensitive and less toxic, aiming to overcome these limitations in patients with severe refractory lupus. This first-in-human trial enrolled 5 patients suffering from severe treatment-resistant lupus and kidney inflammation. They received a single infusion of YKS-109, a specially engineered STAR T-cell therapy targeting CD19-expressing cells. The main goals were to assess safety and efficacy, specifically looking at the SLE responder index for SRI-4 at 3 months, with continued monitoring for 6 months. Patients discontinued other immunosuppressive therapies before treatment. The results were highly encouraging. All 5 patients achieved the primary efficacy endpoint, the SRI-4, by month 3, and this response was maintained through month 6. Crucially, the therapy proved safe, with no severe adverse events observed, including no signs of graft-versus-host disease. While B cells were rapidly depleted, they reconstituted within a couple of months. 2 patients even achieved complete renal remission. This study provides compelling evidence for the safety and potential effectiveness of this novel hypoimmune STAR T-cell therapy in patients with severe lupus who haven't responded to other treatments. A key finding is that these engineered cells disappeared relatively quickly, which could theoretically lower the risk of long-term complications. These promising results lay the groundwork for further investigation of YKS-109 in lupus and other autoimmune conditions. Up next, our second paper out of 5, S12 Micro, a machine learning-derived serum miRNA panel for high-performance pan-cancer diagnosis via liquid biopsy by Zining Long and Jiaming Liu from A Department of Andrology, Guangzhou First People's Hospital, South China University of Technology, Guangzhou, Guangdong, China. Let's break down its key contributions. Cancer remains a major global health challenge, and early detection is crucial for improving patient outcomes. While biomarkers like microRNAs have been identified, their clinical utility for widespread diagnosis is still under development. This research focuses on creating a new, non-invasive diagnostic tool using a panel of 12 microRNAs found in serum. To develop this diagnostic panel, researchers analyzed serum microRNA data from over 20,000 individuals, including healthy controls and patients with various cancers. They employed machine learning techniques, testing numerous algorithms to identify the most effective 12 miRNA signature, which they named S12 Micro. The study rigorously validated this panel across multiple datasets to assess its diagnostic accuracy and explored potential biological mechanisms. The results were highly encouraging. The S12 Micro panel achieved an impressive mean AUC of 0.987 in large validation datasets, indicating excellent discrimination between cancer patients and healthy individuals. In smaller validation cohorts, the AUCs range from 0.900 to 1.000. Furthermore, the panel showed superior average performance compared to previously published serum miRNA signatures and demonstrated technical feasibility through RT-qPCR testing. In conclusion, S12 Micro represents a significant advancement as a potential non-invasive tool for diagnosing multiple cancer types using a simple blood test. While these findings are promising, further validation is essential. Future studies should prospectively evaluate its performance in real-world settings, specifically its ability to distinguish cancer from benign conditions, and ensure consistency across different laboratory platforms. Up next, our third paper out of 5, discovery of potent and highly selective sodium-dependent glucose cotransporter 2, SGLT2, inhibitors for treating diabetic nephropathy by Hao Yanai and Fengjin Wang from Department of Pharmacy, the affiliated hospital of Suzhou Medical University, Suzhou, Jiangsu, China. Let's break down its key contributions. Diabetic nephropathy is a serious complication of diabetes, and its prevalence is alarmingly on the rise worldwide. A key strategy to manage this condition involves targeting the SGLT2 protein in the kidneys, which reabsorbs glucose. By blocking SGLT2, we can reduce glucose levels and potentially protect the kidneys. However, it's crucial to ensure that any inhibitor is highly selective for SGLT2 to avoid unwanted side effects associated with inhibiting other transporters like SGLT1. To find new drug candidates, the researchers employed a multifaceted approach. They started with sophisticated computer-based virtual screening, combining several techniques to predict potential inhibitors. Promising candidates were then tested in laboratory experiments using cells expressing human SGLT1 and SGLT2. Finally, the most promising compound, dubbed HIT4, was evaluated in diabetic mice to assess its effectiveness and safety in a living system. The results were very encouraging. HIT4 showed remarkable potency, inhibiting SGLT2 with an IC50 of just 1.5 nanomolar. Even more impressively, it exhibited an exceptional selectivity ratio of over 3,500-fold for SGLT2 compared to SGLT1. In subsequent studies with diabetic mice, HIT4 effectively lowered blood glucose levels and increased the excretion of glucose in the urine, indicating its therapeutic potential. This study successfully identified a promising new compound, HIT4, which is highly selective for SGLT2. This selectivity is key, as it suggests a potentially safer treatment profile for diabetic nephropathy by avoiding the side effects linked to SGLT1 inhibition. While these findings are exciting, further research and clinical trials will be necessary to determine if HIT4 can truly become a valuable therapeutic option for patients. Up next, our fourth paper out of 5, Antisense oligonucleotide-mediated knockdown therapy in 2 infants with severe KCNT1 epileptic encephalopathy by Tojo Nakayama and Timothy W. U for Genetics and Genomics, Boston Children's Hospital, Boston, Massachusetts, USA. Let's break down its key contributions. KCNT1 related epileptic encephalopathy is a devastating condition affecting infants, characterized by severe, intractable seizures and profound developmental impairment. It's caused by specific genetic mutations that make brain cells too excitable. Sadly, existing treatments offer little relief, and many affected children do not survive past early childhood. This research explores a novel therapeutic approach to target the root cause of this devastating disease. To address this challenge, researchers developed an antisense oligonucleotide, a specialized molecule designed to reduce the production of the faulty KCNT1 protein. This therapy was tested in 2 young girls with a severe KCNT1 mutation. They also used patient-derived cells in the lab to understand how the therapy works at a cellular level, focusing on its ability to correct the underlying electrical abnormalities in neurons. The results were promising. Both infants experienced a significant decrease in the frequency and severity of their seizures. In a lab, the ASO treatment successfully corrected the abnormal neuronal firing pattern seen in patient-derived cells. However, a concerning side effect emerged, with both patients developing hydrocephalus, indicating a potential toxicity that needs careful monitoring. This study represents a significant step forward, demonstrating the potential of ASO therapy for a previously untreatable genetic epilepsy. While the clinical benefits are encouraging, the observed hydrocephalus highlights the importance of careful safety monitoring and further research. Future work will focus on refining this approach to maximize efficacy while minimizing risks, offering hope for children with this devastating condition. Up next, our 5th paper out of 5, tau oligomerization induces nuclear lamina invagination and chromatin remodeling in Alzheimer's disease by Shuo Yuan and Lulu Jiang from Neuroscience, University of Virginia School of Medicine, Charlottesville, Virginia, 22908 USA. Let's break down its key contributions. Alzheimer's disease is a devastating neurodegenerative disorder, and the abnormal aggregation of tau protein is a key hallmark. While we know tau pathology progresses, the exact mechanisms driving neuronal death are still being uncovered. Recent studies have noted changes in the nuclear membrane, but it's been a mystery whether this is an early event or a consequence of the disease. This research aims to shed light on this by exploring the direct link between tau and the integrity of the cell's nucleus. To tackle this question, The researchers employed a multi-pronged approach. They examined post-mortem brain tissue from Alzheimer's patients and a mouse model that mimics tau pathology. Crucially, they also developed a sophisticated system in human neurons using light to control tau aggregation in real time. This allowed them to directly observe the effects of tau oligomers on the nuclear envelope using advanced microscopy techniques. The findings were striking. The study revealed that disruptions to the nuclear lamina, the structural support of the nucleus, were present even in the early stages of tau pathology in human brains. They demonstrated that toxic tau oligomers directly interact with key nuclear proteins, leading to physical invaginations of the nuclear envelope. This structural damage was accompanied by changes in how DNA is organized and regulated, suggesting a cascade of cellular dysfunction initiated by tau. So, what does this mean for Alzheimer's research and treatment? This study strongly suggests that problems with the nuclear membrane aren't just a side effect, but an active contributor to neurodegeneration in AD. This opens up exciting new avenues for therapies, potentially by developing drugs that protect the nuclear envelope or prevent tau from interacting with it. While more work is needed to fully understand these complex mechanisms, this research provides a critical new piece of the Alzheimer's puzzle. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.